Today I will show you how to prepare retinal slices of the tiger salamander for patch clamp recordings. First, I will prepare two kinds of microscope slides. We use slides that have an adhesive well attached to them so that we can use them as fluid chambers. On the first slide, I place two parallel bands of vacuum grease. I then take some filter paper and cut it into rectangular pieces with a razor blade. Now I place one of the pieces on the grease rails and push it down so it's well attached to the slide. I do the same for the other piece. The second kind of slide has little rubber blocks glued to it.